Cool. All right, I'm going to share my screen here. Welcome. I'll be looking kind of like at a weird angle because my camera is down here and my presentation's up here. So I apologize for that. I hope you're all having a great Balticon so far. Okay, hold on. Let me share the screen. Open this up. Okay, so this is how this is going to work. I'm going to talk and you're going to have questions, hopefully. There are a few natural breaks in my talk, and then I'll go in. So if you have a question about whatever, put it in the Q&A. That's the option you should have somewhere in your screen where you can like type in a question, and I'll look at that periodically and try and answer any questions as they arise. I know that there's also the chat feature. It's hard for me to monitor that while I'm talking, so I'll try and look at there too a couple times, but that's more for if you guys are just doing your thing and talking, that's fine. And I'll kind of go back through there after my talk. But the Q&A, I'll probably every like 8 or 9 slides, there's a couple section breaks, and I'll go back and take a look and try and answer as many questions as I can. And then there'll definitely be some time at the end. So welcome. So my name is Dluzen. I am a geneticist, and I am an assistant professor of biology at the department, or I'm in the Department of Biology at Morgan State University, which is right in the heart of Baltimore, Maryland. If you have any, want to follow anything I do, my Twitter handle is ripplesintime24. I post a lot about science communication. My talk today is bacteria and the microbiomes in homes around Earth and above Earth. And so I'm really hoping just to share a little bit of what I've learned about the microbiome. And so the caveat to my talk is that I'm a human geneticist, trained to study health disparities and how social determinants of health influence our health outcomes, particularly with Baltimore City. But I got really interested in a microbiome when I was in graduate school, and it's something that's been kind of a tangential hobby of mine that I've really dived into with a new project in my laboratory now. And one of my graduate students is spearheading a project where we're going into different homes in Baltimore City and collecting mold samples and profiling the types of mold that are in people's homes and whether they're pathogenic, what's there, what kind of bacteria are living there with it. And that really got my curiosity about the microbiome. And so this presentation is kind of a overview of what I've learned about the microbiome coming from someone outside of microbiology. So there might be specific questions about microbiology that I will not be able to answer. And I'll definitely be as forthright with that as I can. Or I will at least try and point you in the direction of who might know those answers. But hopefully I can answer as many questions as possible. So without further ado, let's begin. Let me know if there's any technical issues with the slide. Okay, so a simplified tree of life. So if we start at the origin of life, the first cells that arose out of that primordial soup billions of years ago on planet Earth, and we look at time here as the y-axis, at some point there was a divergence, and that most of the current species of bacteria arose from some common ancestor that also gave rise to us. We're within the eukaryotic kingdom of life. And somewhere along this branch, this is contested. It is unknown if kind of the eukaryote derivatives began first, and then archaea, which are other type of single-celled organisms that are a lot like and similar and dissimilar to bacteria, might have splintered off, or if it's actually the opposite, or if it was really bacteria and archaea that began and eukaryotes kind of splintered off. This is a very simplified tree of life, and I'll talk a little bit about how we know this tree of life is how this arose. But this more complicated picture is the current tree of life. And highlighted in red here are where we fall in the animal kingdom, in eukaryotes, all other, yeasts, even fungi, these kind of things all fall in this area here, which is when you look at the diversity of species on planet Earth, we are similarly related to archaea bacteria, and more so than just normal bacteria, but there are so many more bacteria on planet Earth than there are us, and there are other types of eukaryotes. And so this is kind of a weird kind of picture representation of the tree in life, kind of a more classical example of looking at how everything's related to one another evolutionarily is to do something called a phylogenetic tree. This is a representation of knowing the genetic sequence of a species genome. So like our genome, the genome of bread yeast, the genome of Helicobacter pylori, a bacteria that cause ulcers in the stomach. And we can compare their similarities and dissimilarities, and through that build a relationship tree that you see here. And again, highlighted in red are the eukaryotes, and you can see that we're really just a tiny slice of the pie of everything that's out there. Certainly, we are a dominant species on planet Earth. We are by no means the most populous of species on planet Earth. In fact, as we'll learn in a few minutes, human cells are not even the most dominant type of cell type that encompasses who you are in your body. But we fall in kind of this smaller area here of archaea, bacteria, and bacteria. So I wanted to show this just to get an appreciation for what it is that the diversity of life looks like. So we had a question of the current tree of life. Where is the base? And the base is two places. One, the base is kind of just here, and the base is some primordial ancestor that we don't know what it was, but the original cells, or maybe even the original cell on planet Earth, we don't know if it arrived here from an asteroid, from another planet. We don't know if this was something that spontaneously kind of constructed itself from macromolecules in the early part of our history on Earth. But it's some ancestor that then developed into a cell that was capable of self-replication, that was capable of performing enzymatic reactions, of obtaining energy from the environment, and storing that information so that it could be passed on to the next generation after cell division. So the base of that keeps getting pushed back a little bit further in our history evolutionarily, but we don't know exactly what it was. And so that is determined right here, is that we know that some ancestor, given the fact that even though we're so different from bacteria, we still have a lot of commonality. We share genes that are 100% identical nearly with trees. And we share genes between trees and us that are extremely similar to bacteria. So we know that there must have been some kind of original, OG bacteria that kind of gave rise to all of it. Okay. So when I'm talking about the three domains of life, there are a couple of things that I'm not going to get into too much specifics on this table, but really we have these three major domains, and some microbiologists may even argue that this is an oversimplification. I'm not going to go into that. But there's bacteria, archaea, and eukaryotes. And so these are the major distinctions between the two. The most important one is that the nuclear envelope. So in our cells, we have a nucleus, and that's where our genomic information is contained. We have that as a eukaryotic cell. Both bacteria and archaea are missing that. They have kind of a region within themselves where that information is stored, but there's not another envelope around it. There's not another packaging around that information to keep it safe and to keep it from being damaged. There are different types of organelles. So we have mitochondria, we have lysosomes, many other organelles within our cells that do different functions that you may have heard of. They're membrane bound as well, just like the nuclear membrane, and absent are gone in bacteria and in archaea. The cell wall, how the outside of the cell protects the contents of the cell and interacts with the environment, has different components in it. And if you look at that between bacteria, archaea, and eukaryotes, we're missing that, a protein called peptidoglycan. Bacteria have it, and archaea and eukaryotes don't have it. Protein synthesis starts with an amino acid in the archaea domain and the eukaryotic domain called methionine, whereas it's a little bit structurally different for bacteria in an amino acid called formylmethionine. So some of these kind of hydrocarbon chains, these lipids, are a little bit different and unique to each domain of life. Some of them are very similar. You'll see here that we're more common with bacteria in our membrane lipids than we are with archaea when it comes to membrane lipids, so unbranched hydrocarbons. So there's a lot of traits. I'm going to go to the next slide here. If you look at the genes themselves in our genetic information, bacteria don't have introns, and intron is a part of a gene that kind of gets edited out before the gene is its final product and translated into protein. Archaea have some of these introns, and we have many of them. And in fact, all of our genes have intronic regions, which is kind of like structural components of the sequence that don't necessarily code for protein. So I'm not going to go through this whole table, but there are some commonalities and differences that have provided good evidence to suggest that we're probably more closely related with archaea than we are bacteria. However, all three of us are all linked in that we all stem from some original progenitor type of cell or macromolecule billions of years ago. So I just wanted to kind of give a little bit of an overview of that before diving into the microbiome in general. And let me just check real quick. This is a good little pausing point. There's a branch of the Chiavite that is simply labeled Loki Thor. I believe it, and it's probably due to the fact that the people that discovered it either wanted to call it something cool or wanted to have a little bit of fun. So for instance, there are some gene names like Sonic Hedgehog and a couple others where they were discovered in the 80s and 90s. And the scientists that discovered the gene were able to give it kind of a fun name. So there's a couple of those, particularly in mice. The gene names are quite hilarious. Let's see here. Okay, next slide. Okay, so that's really obviously a brushstroke of types of organisms on planet Earth. And so my talk is really centered on something called the microbiome. And what do I mean by that? I thought maybe just a kind of a basic definition to work from for this talk would be helpful. The microbiome is really the genetic and cellular material of all microbes in a given location. And I'll come back to that in a moment. So that would include, single cellular small microbes like bacteria, archaea cells, fungi, particularly those that are single celled, protozoan, those are eukaryotes, but they're single celled, and viruses. Now, viruses are not a living thing. The definition of being alive, part of that definition means that you are capable of providing the information for yourself to self-replicate to the next generation. Viruses must have a host cell, as we've all probably learned through COVID and a lot of the other viral pandemics, that a virus must infect a host to be able to hijack the host machinery in the cell to replicate itself. So by definition, it cannot self-propagate, which means it's not truly alive. However, when we're talking about the presence of microbes in the microbiome, we include viruses in that definition. So it can be a little bit tricky sometimes when we're talking about things that are truly alive versus not, but we do consider viruses as part of kind of the microbiome. And today's talk is really going to focus on several different things in the microbiome, and that's where I go back to given location. Today's talk is really going to focus on what's going on us in humans, the microbiome-wise. Why should it matter that we have bacteria on our bodies? Why do we care? Why is it important? And then we're going to talk a little bit about the microbiome in the world that we interact with, outside in the soil, on our walls, around the home, where we might pick up the microbes. And then we'll talk a little bit about space, which of course you can see behind me. I'm definitely on the International Space Station right now, which will feature later on in our talk. All right, so, okay, we'll come back to questions in a minute. All right, so before I get into that, one thing we want to talk about, or I want to talk about, is how do we know what's there? How can we look at something that we can't see? And certainly we could take a culture of it and look at it under a microscope, but that doesn't really tell us if it's, for instance, an archaea or a bacterial cell. The subtle differences are very hard to see under a light microscope and sometimes even a confocal microscope, which is a little bit more higher powered with higher resolution. So the discovery of archaea, for instance, didn't really occur until the 70s, when we had the technology to extract someone's genomic information and sequence it, like reading a book, and understand the precise orientation and precise lettering of the genome. Our genome is consistent, and our genome of everything on life, even in viruses that aren't alive, have a genome that is made up of 4 bases, A's, C's, G's, and T's, adenine, cytosine, guanine, and thymine. These are 4 acids, basically, nucleic acids. And there are several different ways we can extract genomic information and read the letters of the code. The original method was called Sanger sequencing, where you kind of built a template one by one. I'm not going to go into the specifics of how this works. It doesn't matter for this talk. But basically, we have developed technology now where we can take, say, our cellular genomic information, and we can read out exactly what DNA is there, and not only in what quantities, but what's the precise sequence of it. Now, there have been revolutions in this technology. So another technology is called whole genome shotgun sequencing. The reason why I'm bringing this up is that you can take a sample from the environment that has viruses and bacteria and fungi all kind of living together in one complex ecosystem. We can take their genomic information, you break it up into little bits, you sequence all those little bits, and then you use computational biology to restructure that all back together and identify the different types of species that were there. So that's really all I want to get into with sequencing. There are definitely more advanced technologies now that provide better resolution, different things that it doesn't really matter for this talk. And I'd be happy to talk about a little bit of those, at the Q&A if you'd like. But this is really what allowed us to really identify this species of bacteria versus this species bacteria. And it's all based on that phylogenetic tree I looked at earlier, which is, how closely related are these species? Are they the same species or are they different? And how different are they? And that really allows us to classify then when we have a new sample, an unknown sample, sequence everything that's there and then compare it back now to the databases that we have of all of these different microbes. So let's get into the human microbiome. So what is going on and inside our bodies? And I thought this would be a good jumping off point because we all care about ourselves and we're also kind of battling the COVID right now and other pandemics that are occurring and common cold even. So what's going on here? So there was a study a couple of years ago. There have been a few studies, but one of the most recent ones was a few years ago. And that was how many cells are there in a human body? And so this is looking at a typical adult female, I believe. And in a typical adult female, you have about 25 trillion human cells. And I'm sorry, about 30 trillion human cells. 25 of them are your blood cells, 25 trillion of them. They take up about 3 kilograms of your mass of a typical adult individual. Erythrocytes, so those are blood cells. Fat cells, adipocytes, take up another good chunk of portion of by weight of your mass. But really, when we look at like erythrocytes, they are the most type of cell we have by a wide margin. And then everything else, all of our tissues, the other 5 trillion or so cells in our body that are human cells, are our neurons and muscle cells and all these other cells. Bacterial cells. Now, this doesn't even count the amount of virus on you. This doesn't count for the amount of fungus that might be on you or yeast. Account for about 38 to 40 trillion cells. These line your gut. They're on your skin. They're in your mouth. They're in any mucus layer inside your ear, in a vagina, on your toe. They're everywhere on your body and inside your body and all your tissues and floating around in your circulation in the blood. When we look at that by weight, our human cells definitely outweigh the amount of cellular content from bacteria. But when it comes to just pure numbers, you're mostly bacteria, which I think is kind of a fun thought. And most of that is a very, what we call symbiotic relationship. We are working with bacteria and providing them with something, and they're providing us with something. And that's kind of what I wanted to get into some of those somethings. So one great place that you might have heard about, you eat yogurt and you can help your gut flora, but your gut microbiome. I'm going to take this. It's very warm in this room. So our microbiome provides us enormous health benefits. That also means that if something messes up with our microbiome, we can have disease and health consequences. But this is kind of the typical, it's a very rudimentary drawing of our gut, and we call it the gut lumen, which is if you were the inside of your small intestine, inside of your large intestines. Exactly. So this is why antibiotics can make you sick. Thank you, Jennifer. Right. So we can, well, I'll come back to that point in just a second. So we're eating our food, and from our food we get protein, fats, and fiber. These are necessary nutrients for the bacteria, which are kind of almost the first layer of interaction that our nutrients get from our food before it processes in our body. These bacteria help us process these foods and these biomolecules. So fats get metabolized into primary acids and secondary bile acids. These are essential for our digestion. There are some things that we need every day that we only get from our food, and that's only produced by the bacteria in our gut. Fibers, importantly, certain types of bacteria called firmicutes and bifidobacteria create butyrate and folate, and these are biomolecules actually that if you have higher portions of them in your diet, this reduces the mucosa of your intestines. So these are our actual intestine cells from dividing uncontrollably, and it actually reduces our cancer risk, which we'll come back to in a few minutes. Whereas if you have more of a high protein, high fat diet, the bacteria there are going to be creating hydrogen sulfides, secondary bile acids, which can actually trigger inflammation and cellular growth and proliferation in our guts and increase our cancer risk. So the balance between good and bad bacteria is more about a lot about the food you eat and the types of nutrients these bacteria process, but they're absolutely essential. If you were to wipe out all the bacteria in your gut, you would be feeling very sick. And so that kind of goes back to Jennifer's comment that like if you overtake antibiotics, that's sometimes why you get diarrhea. It's sometimes why you get GI problems, is because those antibiotics are killing whatever it is that you need them to kill, but they're also killing good bacteria that kind of provide the health benefit that are going on in your gut. Just gonna take a look at the questions and answer. We will talk about the non-pathological human virome later on. Any thought on good bacterial supplements? I'll get back to that at the end of the talk. Are there diseases caused by archaea? There are. I know there are. I don't know offhand. Let me think about that while I'm talking. I don't know offhand, but I'm pretty sure that there are. And yes, C. diff infections are definitely an opportunistic bacteria that generally most of us have C. diff in our bodies, almost all of us. It's like most of us actually have staph on our skin. It's more of an opportunity. So if you take a lot of antibiotics, like if you get sick and you go to the hospital and you're on antibiotics for 3 weeks, you either get exposure to more C. diff, or that C. diff now has an opportunity to grow and expand in the gut lumen and in the gut area where those other bacteria have died from the antibiotics. And so it's kind of a game of catching cat and mouse, where you want to kill the good bacteria or bad bacteria quickly, and you want to let the good bacteria recover quickly before the bad bacteria that didn't get killed by antibiotics develop quickly. So it's kind of an interesting game that goes on in your gut. Okay, now the human skin microbiome. So we have a whole layer of dead skin cells on our skin, and then on top of that, we have a lot of different bacteria. We have viruses that interact with this bacteria, phage viruses in particular, which are viruses that only infect bacterial cells, and then fungus and several other types of microbes. Well, the recent studies have shown actually that the skin microbiome is relatively stable for us. So even depending on whether, how often you're bathing, who you're interacting with, certainly you might pick up certain types of species from people that you haven't had, but relatively speaking, if you look at different places, say on your face, on your arms, on your foot, in the crux of your elbow, those stay relatively stable once you're an adult. However, in childhood, that's a little bit different. So I'm actually going to skip ahead a slide and I'll come back one. Most of our gut microbiome and some of our skin microbiome is actually seeded right at birth. So for instance, there was a recent study looking at mother-child pairs. They looked at about 50 pairs, and this has been replicated now in a couple other studies. But you take an oral swab of the mouth, you take a skin swab, a stool swab, and then a vaginal swab of the mother. And then on day one, and then following up months after the child is born, they take similar swabs. And what you find is that when you look at the oral swabs, a lot of the oral cavity is predominantly seeded by what you get from mom. And our stool, which is when you collect stool and feces, this is a representation of what's going on in your gut. It's not the representation, but certainly anything that's being processed out of the body. Find that our gut microbiome in particular is highly reflective of the stool samples coming from mom. I'm suggesting that a lot of the bacteria you pick up originally with you come from mom, which I think is also really great and also aids in our digestion. Colostrum and breast milk also have bacteria in them and also help seed some of the gut bacteria, though that was not included in this study. I'm going to go back a slide. As we age, though, unlike our skin microbiome, our gut microbiome changes a lot. Certainly with diet, if we look at, say, levels of bifidobacteria, we have much higher levels of those early in life, generally seeded by adults and then by your parents or your mother in particular. And as we age into adulthood and then into old age, we lose out on a lot of this bacteria that are generally considered to be pretty healthy bacteria and bacteria that are beneficial for our metabolism. And so as we get disruptions in bifidobacteria or other types of bacteria, we can be impacted, have allergies. Irritable bowel syndrome is certainly a condition that people suffer from that is very much linked with the microbiome in the gut. Obesity and diabetes is also linked now with the metabolism in our gut. We'll talk in a few minutes about some of the other health benefits of the gut and cancer risk, particularly colorectal cancer, stomach cancer. Some of those other cancers have direct links and risk factors associated with the types of bacteria that you either got from the food you were eating or the types of bacteria that have found their way into your gut and are now kind of spreading. Of course, this is kind of generalization. This is not true for any unique individual. This is pooled samples looking at over time as in individual ages, and certainly is going to be related to where you live. So this is a study looking at the bacterial content of the gut microbiome. I'm just going to check my time, make sure I'm doing it. Okay, I gotta go a little faster. The gut microbiome as influenced by diet. So this is looking at all the bacterial content of African Americans in the United States and Native Africans that are living in South Africa. Most, or at least I should say, in this study, the African Americans that were recruited eat a high-fat, high-protein diet, whereas the Native Africans recruited in this study at more of an agrarian fiber-containing diet. And here you can see, this is kind of across all the individuals in the study, you can see different degrees of percentage-wise of different types of bacteria. It doesn't really matter. The main take-home point is that bacteroids are bacteria that really process fat and protein and provide those inflammatory metabolites that get into your body. Whereas Prevotella and those related bacteria, this is a phylum of bacteria, are much more likely to be developing certain folates and butyrates, which are metabolites essential for our body, but also keep inflammation down and lower cancer risk. This study was interesting because they took these individuals and switched the diets, and over 4 months found that their gut microbiome also switched. And then when they went back to their normal diet, the diets reverted back to what they had originally been before the study. So this was highly, highly related to the types of foods that you're eating. This was a study looking at individuals in Nepal, 4 different populations, and then the US as a comparator. Looking at individuals that are really on a hunter-gatherer diet, individuals that are remote, moving from area to area as they follow their food and collecting and foraging. More settled foragers that have kind of established communities, have some agrarian practices, and then are also going out and collecting food, and then more agriculturalists and those that are settled in cities in Nepal and Kathmandu. And then they classify this as industrialists or more like an economically developed country like the United States, and looking at different types of bacteria related to the diet. And you see that some bacteria, particularly in Nepal, are much more closely related geographically than those in the United States, whereas you see some subtle differences in the types of bacteria that are found in the diet, particularly in Microbia. These are types of bacteria that help with our diet and processing. So our diet is heavily influenced by, our gut microbiome is heavily influenced by our diet. All right, so now I'm going to get into the built environments. This is a good little pause here. Let me just kind of catch up with some of the questions, and let's see here. I'll come back to bacterial supplements. So no transplant patients taking immune supplements are told to change their sheets more often and wash pajamas more often to avoid a buildup of normal skin staph that might turn into dangerous infections. Yes, so I mentioned earlier that staph are on our skin. We all have staph. So we all have staph that are capable of causing staph infections. It's just whether or not that staph gets into an open cut or a wound to cause a staph infection. Certainly you can pick up staph from the environment, particularly MRSA, which is a methicillin-resistant staph, and highly drug-resistant staph you can pick up in a hospital. But Absolutely, we all have staph as a natural component of our skin flora. Great questions. Okay, so we can pick up our microbiome from many different areas. Certainly, ventilation in your house. If you have pollen blowing in, for instance, you might be picking up some of that bacteria. That's actually how penicillin was discovered. There was an open window in the laboratory, and Alexander Fleming found that the fungus had come in and landed on one of his bacterial plates and produced a natural compound to kill that bacteria. And that was how the first antibiotic was discovered, very serendipitously. We can pick up microbiomes that are reflected in our pets. There are several studies that have looked at the gut and skin bacteria of your pets and animals and from their dander, and we find that it's on our skin and in our gut. Same if you have cockroaches or other types of pests in the home. Farmers and those that are exposed to livestock or horses on a routine basis will pick up some of it and share some of the same bacteria and microbes that are found there too. So there's a lot of different ways we can pick up the microbiome. A really interesting study was looking at the microbiome of handrails at Penn Station in New York City. And so this is a study done by Chris Mason at Cornell University, where they went and just sampled every hour for the day the handrails. They took swabs of the handrails and then did sequencing on them to find the bacteria there. And they found that not only were there all kinds of different bacteria, viruses, they picked up a couple pathogenic stuff too, but that there was kind of a temporal change in how often these bacteria were changing, probably due to rush hour. So 3 o'clock, this is the beginning of the afternoon, here's the morning. And you can just see over time, the microbiome of just a stationary object at Penn Station and those handrails changes, certainly influenced by the people touching those areas, and then definitely over time, the amount and frequency of those touchings. So they kind of created almost like a clock, and actually his study, they ended up taking swabs from every rail station in the whole city. And there's a lot of other interesting stuff in there. And the citation is down on the bottom right. And we can probably figure out a way to share these slides or something if you're curious of following up. Where are the microbes coming from in the built environment? If they're coming from us, this was a study looking at homes in Brazil. And we find that, for instance, in our bedroom, most of the microbiomes in the bed, so that you leave that slough off your skin or that you breathe out and settle on to your pillow at night, come from your human arm and also our feet. Same with chairs and countertops. Cups also come from water sources in those homes. And you can find definitely in the floors in the bedroom, the kitchen, and the living room. Most of it's our feet tracking in. Walls in the bathroom, in particular, the oral microbiome influences. So that's identifying that there are bacteria in the walls in the bathroom that are found there and only there in the house, and also in the mouth of the occupants. So this is probably due to brushing teeth, showering, cleaning yourself, that you're exposing and dropping out these bacteria, and they're getting onto the walls of the area. So the built environment is definitely a reflection of the occupants, and a reflection of the occupants going out into the environment and interacting with other things and then bringing that back in. Certainly there are a lot of undertones for COVID. I had originally wanted to talk about COVID, but I realized I think there's just it was an easier escape for me to put together a presentation that didn't deal with the pandemic. So I thought I can answer any questions about COVID afterwards. I'd be more than happy to. So I thought this was a very interesting study. Certainly in our kitchen and the walls of our kitchen, heavily influenced by tap water coming in or cooking water sources that also are human skin. So we're touching our walls, we're moving things around, touching the countertop, and that is definitely changing the kind of the local environment of the built environment. Okay, so I'm just check questions real quick because we're going to get into the last part of it. I'm going to try and get this done in about 10 minutes. I'll leave a couple answers, questions and answers. We can use sequencing to profile then the microbiome of things all around the world. So this is a study that just came out a couple of years ago looking at bacterial diversity around the Earth. And so they went and took soil samples from all the continents except for Antarctica, and in different climates and biomes of these continents. And they found a whole host of different types. So this is looking at the different relative abundance of bacteria by phylum breakdown. So proteobacteria. And there are different types of proteobacteria, predominantly around the world, most are alpha proteobacteria. I don't really know what that means. They're a type of bacteria that metabolize certain things. I'll be honest, this is more the extent of my knowledge about these specific bacteria. Actinobacteria are definitely bacteria that we have on our human skin, and they're pretty prominent actinobacteria in the environment. But we can see that when you break this down, there are certain types, so like certain types of bacteria that are more predominant in temporal and tropical grasslands. Proteobacteria are more common in forests and in cold forests. Dry forests have more other types of actinobacteria and firmicutes, which I'll come back to in a minute. Tropical forests have their own types of bacteria as well. Some of these, when you sequence these, not only do you identify the species, but you can identify genes that give you a little bit of an idea of their functionality. So there are certain types of actinobacteria, acidobacteria that are very good, say, in areas of, when you look at ecology of low productivity. So deserts, deserts in particular, or area wastelands that don't have a lot of metabolism going on nutritionally. These are bacteria that will thrive there. There are bacteria that thrive pretty much everywhere in the world, from the vents undersea of the ocean to volcanic lakes in Glacier National Park. Some bacteria thrive in low pH acidic environments. And so you can kind of identify from the types of genes in these genomes where or what the functionality of the metabolism of these bacteria are. And when you map that to the world, we find that a lot of, say, high pH bacteria, so these bacteria that live in basic areas, and when I mean basic, I mean basic alkalinity, as opposed to high acidity. Those are areas that are very desert, like in Mongolia, the high steppes of Central Asia, the Western United States, or the Atacama Desert, or in the Sahara, areas that don't have a lot of moisture. Whereas in dry lands, other types of bacteria are present, particularly in the Sahara. And then other areas, especially arid, dry areas that kind of overlay with some of these other areas, we find that there are low productivity bacteria. So bacteria that can kind of thrive in this type of environment. And the last part of my talk is going to talk about the link between all these creatures. So bacteria, archaeoviruses, they have a very intimate history. For example, for childbirth, the placenta are actually, there are genes related to the formation of placenta that are originally found in viruses and originally evolved in viruses. So we even have some genes in our genome that we've hijacked from viral genomes. That's no different when you look at bacteria and viruses. They are constantly at war with one another, and they're forever shaping each other's evolution. And that is probably one of the driving factors that caused archaea and eukaryotes to splinter off from bacteria. Bacteria and viruses can exchange genetic information. And so this will allow, say, a phage virus to pick up, to say that it infects a cell, bursts the cell open, that exposes all the DNA from that cell, and other bacterial cells that are kind of just locally hanging out there, maybe even from a different species, can pick up that information. Other times, viruses can actually go into a cell in the bacteria, pick up the cellular genomic information of that bacteria, leave that cell, and then go and infect a different species and transfer that DNA into that new bacteria. This is a primary driver of evolution as we know it, and also a way that information can be exchanged between different kingdoms of the and different domains of the tree of life. And so this intimate history has really driven a lot of things that we've seen when it comes to the different types of bacterial microbiome organization. I'm going to skip this slide. I think this is really interesting is that, though, when we look back at our gut lumen, so here's our gut, here are the mucosal layers of the bacteria, and then here are some more bacteria, and the exchange of information. That pattern of structure that allows information to come from nutrition into our bodies is very similar when you look at, say, seawater and coral, or you look at sea anemones and other types of flora in the sea, where they exchange nutrition with seawater. They also have a bacterial layer protecting their tissues that exchange information. This is also true in soil, where plants are picking up nutritional information from the soil. It's being processed by bacteria and other types of microbes and then brought into the tree itself. This all independently evolved. It's called convergent evolution, where these similar types of pathways evolved which really goes to strengthen the argument that this is an essential feature of life, an essential feature of how we interact with the microbes around us. And so I actually discovered this looking for this talk. Okay, Tim and Oren, great, thank you. And I thought that that was really interesting. Okay, so I promised space. Let's talk about space. The microbes of the built environment at APL, Applied Physics Laboratory, or JPL, or at NASA, or JAX, or some of the other major space organizations around the world, they do as best a job as they can at decontaminating the information going up into the ISS and in the space shuttles, but it's not possible to get everything. And certainly then when humans come to the ISS, they're bringing everything that's on them to the space station. And so there have been several studies that have looked at diffusers, handrails, air return grills. This is a picture from the ISS. This was published actually just a month ago. And you can find that when you look at the different types of microbes and bacteria, there are all kinds of things on these different parts of the International Space Station. So it's really a reflection of us. When we look at, this is kind of looking at very large phylum and families of bacteria. When you kind of dive into the species level, Staphylococcus is present in air intakes and in diffusers and in surfaces of incubators. So we can be picking up potentially pathogenic or related types of staph that aren't pathogenic from the environment, even in the space station, which is about as sterile as you can try and be, other than maybe in a hospital setting. So I thought that this was really interesting that we're starting to look at that. And certainly when we go to Mars, when we go to the moon, if we're able to leave permanent colonies there, we're going to be leaving the microbiome there too. There are bacteria that have survived rides around the sun on satellites that have returned back, that have survived zero-G. So I am not at all surprised that there will be bacteria that will eventually find their way onto Mars. Maybe even with the Mars rover there, they've already been seeded on the planet. So it'd be interesting to see what that looks like in the coming years when we can finally get some samples. And then this was looking at dust. So this is on the space station. This was dust particles collected from dust. And you can see that in HEPA filters, when you look at International Space Station on the left, and then the actual center where that stuff was collected, the profiles don't necessarily match up, but a lot of some of the similar bacteria and families of bacteria are certainly carrying through. And it seems like in the space station that then preferentially allows some of these bacteria to kind of thrive a little bit more than others. Certainly like firmicutes on the HEPA filters and then the filters built by JPL and then launched and brought up to the space station can really thrive there. So it's very interesting to see where this is going to go. The NASA Twin study also looked at the microbiome of people in space and how that changes over time. I didn't have time to really talk about that in the talk, but I would certainly, NASA has all that information online. That's the NASA Twins study. So yeah, I wanted to wrap up and just leave about 5 minutes for questions. I really appreciate you coming here. I see there's like over 50 people. It blows my mind. Thank you so much. You can find me on Twitter at ripplesinTime24, or feel free to shoot me an email. I love talking microbiome. I love talking genetics too and aging, and I'd be happy to answer any questions. And then I'll just plug shamelessly that I have several nonfiction articles about the microbiome, about the NASA Twin study, about aging in Clark's World magazine, as well as an analog. So thank you. Let me see if I can pull up any last minute questions here. Okay, so I had promised to come back to, are there any thought on good bacteria supplements? I think that I am not opposed. Certainly certain foods have good bacteria in them. I think yogurts, in particular, kombucha, is it kombucha? Yeah, kombucha, I'm saying that right. Some other fermented foods, I think that those provide some bacteria that can be very beneficial. I don't know anything about things that you find at GNC where they promise that they're going to cure Alzheimer's or something. I think that that's all garbage, but certainly, foods from vegetables. I believe some of these, one bacteria, I'll type it in here, it's called Akkermansia mucifila. Akkermansia mucifila is a bacteria that's been identified as protective against aging-related diseases, and immune system inflammation. So this is a bacteria that produces butyrate and other types of metabolites that get into your bloodstream and actually kind of chill your immune system out a little bit, particularly as you're getting older and you don't want a ton of inflammatory problems going on. There's a discussion in the field of, is it actually that bacteria, or is it some other bacteria that works in concert with Akkermansia that's the real bacteria and driver of this phenomenon, and we just haven't picked it up yet in our studies. And that's an ongoing debate. So there are definitely companies that are now trying to package Akkermansia into chocolate and to eat that. And I don't even want to give an opinion on that yet because it's so early on. I'm sure the science will catch up with that to see whether or not it's right or wrong. Do you have any information about the virome? So there was one slide I skipped over, and I apologize for that. I'll try and bring it up here. That does look at the virome, and part of it is that we just really don't know what any of this stuff is and where it's coming from. But most of it is in the oceans. So most of the oceans contain not only most of the bacterial species, but most of the viruses that are kind of combating those. There's actually an education project I'll type in here called CFAGE, where undergraduates at institutions around the world actually go and take samples in the soil, in the ocean, and sequence it just to figure out like what's there. And so that's kind of where we are with the virome around the world right now, is that we are really just trying to figure out, oh, I'm sending all this stuff to tech. I'm sorry. I should be sending all this information to Peter, maybe you can resend that out real quick. So CFAGE, thank you for typing that in. And then I'll do Akkermansia. Is that we're just trying to catalog it and try and figure out what's really there. So, okay, and then let's see. Does the limited diet analysis cause changes in the astronaut's gut biome, and does that give them digestive problems? Great question, no idea. So I'll be upfront, that's a great question. I am sure that either the NASA twin study looked at that, or NASA has information on their website. They have a ton of stuff about the health of their astronauts, and I would direct you to their websites. If you go to PubMed Central, you can even type in like astronaut microbiome, and it'll pull up papers and abstracts based off of those findings. And I'm sure it's been looked at. I don't know offhand, but you could probably go to PubMed Central and find some of that information. Let's see, probably got time for one or two more questions. Any thoughts on transplanting gut bacteria? Yes. Awesome. It's some of the clinical trials have already shown that's a phenomenal way of treating IBS. It's at least initial studies have shown that a poop transplant, a fecal transplant, taking poop from a healthy individual, a healthy donor, and there are companies now that do this, they collect poop from healthy people, and they transplant that into individuals with irritable bowel syndrome and colitis and gosh, there's one other disease or condition that I'm thinking of that I don't remember the name of. And yes, it's not effective for everybody, but there are a lot of people who report a extreme change in their symptoms, and they are literally getting poop injected up their rectum and seeding new bacteria into their colon. So yes, I'm a big proponent of this. I think that this is a great way to identify new ways of curing these problems that are very hard to cure. C. diff has also been found to be cured with basically priming the colon and the small digestive tract with C. diff with bacteria that kind of compete out the C. diff. So that might be a great way. Another thing on this track is there are people now looking at phage virus as a new type of antibiotic. And so instead of using penicillin or amoxicillin, you actually give someone phage vectors to go in and kill the bacteria. And this was actually used in World War II by the Russians. They didn't have penicillin when it was discovered in the United States during World War II. So soldiers on the front in Russia would actually have a vial of bacteriophage that they would crack open and dump onto their wounds that were infected to kill the bacteria. And so there are definitely clinical trials going on looking at phage therapy as a way of combating multiple drug-resistant bacteria. I'm imagining I'm just about out of time. I'm happy to stay for a few minutes and answer more questions, or do we have to end this, Peter? There's a question, is there a reason whether or not we would want our bacteria to go on Moon or Mars? I think that's from James Pratt. I recognize that name. That's a good question. I don't know. I think that for the science of understanding a unique biome and going into a kind of a purified location, from the scientific perspective, I think then it's a shame that we're contaminating these things. I just think it's going to be, I think it's going to be inevitable anyway. And certainly if we want to colonize the moon and Mars, or even terraform Mars someday, bacteria, there's no way we're going to do it without bacteria. A great example of that is when you take in a breath, all that oxygen that you just took in were created by bacteria about 1 billion to 2 billion years ago. Our oxygen in our atmosphere was a lot different when Earth began, and it wasn't until bacteria started respirating aerobically and releasing oxygen as a byproduct of their metabolism that our atmosphere filled up with oxygen. And that's probably going to be the only way we would ever terraform something like Mars, where you could actually walk outside Mars without an air tank and walk around. And that, it probably took 1 billion years to do that on Earth. Maybe we can up that game a little bit when we terraform Mars, which I hope we do someday. But yeah, that's a great question. Let me make sure I didn't miss anything. Yes. Yeah, Larry Niven writes a lot about that. And gosh, I'm forgetting the author's name, but like Green Earth, Green Mars, Blue Mars, Red Mars, the author escapes me, but there's a lot about the terraforming of Mars and looking at that too. Kim Robinson, yes, thank you. Let's see, CRISPR. Weren't bacteriophage viruses used in an early form of gene editing? Yeah, so CRISPR is actually a bacterial defense mechanism against viruses. And that's CRISPR, as you know, or may have heard, is a technology adopted from bacteria to perform gene editing in human cells. And it's a revolutionizing medicine right now. There are clinical trials right now of editing our own cells and putting them back into an individual to cure sickle cell and cure cancer. So that's going to be, you're going to hear CRISPR in the future in every news line probably in science. But yeah, it's really evolved a long time ago in bacteria as a way of killing viruses. And we've actually found now ways of stopping CRISPR because viruses have co-evolved a way to stop the CRISPR in the cells that they're infecting. So this is like an arms war between Russia and the United States. We're just building better proteins to combat each other during infection of a bacteria. So yes, absolutely. We'll be seeing a lot more about CRISPR. Okay. Yeah. Are there some fungi that have a mutual relationship with their plant hosts? Are there any fungi that do the same for humans? I can't think of any off the top of my head. It would not surprise me though if there were some that provide us some benefit. I just don't know of any off the top of my head. I'm sorry. So yeah, a couple minutes, but yeah, thank you all so much for coming and participating. I'll try and figure out a way to, I don't know if I think this is going to be streamed on their like webcast at like late at night. So if you're up at 4 a.m. and you're dying to hear things about microbiome, it'll be up there. And it'll be on the Balticon website too. And then I'll try and find a way to get those slides linked up there. If not, what I'll do actually is if you follow, I'll type in my Twitter account one more time. If we do that, I'll try and post it through my Twitter account, and then you can download the slides if you'd like. Most of the stuff is all just ripped off from other papers that did way more cooler work than I ever did. So that's my Twitter account. I'll try and put it up tonight or tomorrow morning, and you can just download the slides from my website. Well, thank you. All the great questions. I really appreciate it. That's awesome. And I hope you have a great rest of Balticon.